Welcome everybody, welcome to a new EBD seminar. Today we have a special day. We have two PhD students from the house, both from the Wetland Ecology Department. And yeah, so since it's different, we'll have 30 minutes per PhD student. It will be like more or less 20 minutes presentation, followed by 10 minutes questions. And yeah, all questions please be constructive and only constructive. And first we will have Icíar that did her bachelor in environmental science in Granada, followed by a master's in conservation. And then afterwards we will have Raúl, which is a biologist from the University of Sevilla and also master in molecular genetics. So that's all for me. Just keep constructive suggestions and questions, and that's it. All for you.
Okay. Good morning, everyone. Well, I will wait. And thank you for being here. I'm going to present my thesis plan on seed dispersal by waterbirds under global change. And as you know, seed dispersal is a key phase in plant populations dynamics, since it allows plants to colonize new suitable areas for them. Birds are really good at seed dispersal. It is one of the most important ecosystem services that they provide. And traditionally, it has been assumed that only or mainly frugivore birds were dispersing plants, and specifically fleshy-fruited plants, through a process called as endozoochory under the paradigm of plant dispersal syndromes. However, now we have reasons to not believe entirely in this classification, because this classification is made according mostly to morphological traits. And now we know that waterbirds can disperse a wide variety of plants via endozoochory. For example, in this study, they saw that seeds from more than 400 plant species had been reported in the diet of 31 European waterfowl species. And most of the seeds that are found dispersed by waterbirds are from other syndromes than endozoochory, as you can see in this plot, for more than 300 plant species dispersed by Dublin ducks. And maybe you are only thinking in aquatic plants or macrophytes, but we know that waterbirds can also disperse terrestrial plants, especially Dublin ducks and geese, because of their feeding habits. And to have a seed dispersal event, we also need that seeds to germinate. And since they are not from the classy fleshy-fruited plants, they were not supposed to be adapted to endozoochory. But now we know that these seeds can survive gut passage and germinate. Here you can see the little differences in the seed coat of the seeds after endozoochory by mallards. And in the same study, they tested the differences in germinability between control and past seeds, and they only found significant differences for 2 of the 11 species that they tested. So now that we know that this process exists and that there have been great advances in the last decades, we want to address with this thesis some topics that remain understudied. For example, we would like to see how land use is affecting seed dispersal, since we are performing a great habitat transformations. For example, in the Mediterranean, almost half of the wetlands have been lost since the 70s, and the 23% of the remaining wetlands are artificial. So this transformation can be leading to changes in the habitat use pattern by the waterbirds and also to changes in the plant communities with whom these waterbirds interact. For example, in this study of seed dispersal by grey-like geese in Sweden, we can see the habitat use network by the grey-like geese, and we can see how they visit and connect natural lakes with a wide variety of croplands. So we would like to see what is happening in Mediterranean habitats where, for example, for grey-like geese, there is no any study on seed dispersal. Secondly, long-distance seed dispersal is of vital importance for many key aspects of the biology and ecology of the plants. So we would like to see which role can waterbirds play in this dispersal, because if long-distance seed dispersal events are needed for the plants, the role of waterbirds can be crucial, because if we look at the average maximum dispersal distances that non-fleshy fruited plants could achieve by other means of dispersal, we cannot find quite high values. And apart from the role that waterbirds could have in the actual distribution of the plants, it is also interesting to study which role can they play under the context of climate change, where some plants could need to migrate to deal with an increase in temperatures. This was addressed for fleshy fruited plants and frugivorous birds in this cool study by González Baró and colleagues, and they found that only the 35% of the plant species were being dispersed by northward migrating birds in Europe. And as you know, in Europe, birds migrate to cooler latitudes with this northward migration in spring, and the fruiting peak for Mediterranean and temperate species is mostly in autumn. So that's why they didn't find a strong overlap, and why they said that bird migration can have limited potential to disperse plants to cooler latitudes. However, in the case of waterbirds, this can be different, because they can also feed on the seed bank, depending on their feeding habits. And this can be, so if they feed on the seed bank, we don't need that strong phenological overlap. This can be seen in this study by Orgiana and colleagues, where they collected mallard feces during the whole year, once a month. And they found seeds during all the seasons, but they found a greater abundance of seeds per sample also in autumn, winter, but a higher number of plant species in spring. So with all this information, the question that arises is if waterbirds could help the plants they disperse to keep pace with climate change. And this was explored by Duarte Viana in this study for 3 aquatic plant species, and he found, in short, that the plants can potentially expand fast enough to reach suitable areas in the future, helped by the waterbirds, having expansion rates higher than the local extinction rates. So with all this information, with this background, and having into account that now we can use GPS tracking data for the birds, it will be interesting to see if we can find a correlation between the waterbirds and waterbird dispersed plants distribution, and also if we could predict potential distribution of the plants while accounting for changes in the distribution of their dispersal vectors and for climate change. So the last step for a successful dispersal process is establishment of the plant. And there is very few literature on the effect that gut passage by waterbirds has on it. We can see these 2 studies by Figueroa and colleagues for 2 macrophytes, and they found different results. In the first case, depending on if the plants were exposed to herbivory or not, and in the second case, they didn't find a significant effect of gut passage on plant performance. So we would like to see how is it for non-aquatic plant species, and also what is the effect of the presence of waterbird feces surrounding the seeds on the plant establishment. So in short, a summary of the topics that I would like to address with my thesis is how land use affects seed dispersal, which role can waterbirds play in plant long-distance dispersal, and how does waterbird endozoochory affect plant establishment. So in the first chapter, we wanted to assess the role that wintering grey-lag geese have in seed dispersal in Doñana area. For that, we wanted to identify the plant community dispersed and the differences between habitat and seasons, and also approximate the spatial distribution of the seed dispersed. So for doing that, we first went to the field and collected geese feces, and we extracted the seeds from the feces at the lab, and we identified them using a specific literature and performed germination tests with them. So we collected feces in 4 different sampling sites, 3 natural marshlands and 1 artificial wetlands, who was at Blanco rice fields, and we analyzed 30 samples per sampling site. And this was a project that started with Adrián Monreal master thesis. Here you can see the location of the sampling sites. Here we have the rice field, and here the natural marshes. And you can also see the GPS tracking data we counted with for the study, provided by Anthony Fox, for 3 individual geese. And you can see how the geese were only using Nato Blanco rice fields at the beginning of the season. So with this data, we wanted to approximate where the seeds we found could come from and where they could be dispersed to. So for doing that, we first drew 1 spatial polygon per sampling site, and we selected all the GPS fixes located within the polygon. And then among all the fixes, we selected those within the intervals from 12 hours to 30 minutes before and from 30 minutes to 12 hours later, after each location within the polygons and per individual geese. And the locations selected in this interval are supposed to be the places where geese could be feeding and then ingesting the seeds. So we'll filter them to keep only diurnal locations with a speed lower than 5 kilometers per hour to make sure that the geese were not flying or resting. We chose this time interval because according to this study, the 90% of the seeds ingested by geese are ingested within the next 12 hours. So I can show you some preliminary results. We have found more than 1,700 seeds belonging to 15 different families and 24 different taxa, 7 of them not previously reported as dispersed by waterfall, and the 17% of the seeds tested for viability germinated. And here you can see that none of the seeds we found was assigned to have an endozoochory dispersal syndrome. Here we have the plant taxa relative abundance per sampling site, and these numbers are the total seeds and the total number of taxa found in each of the sampling events. And we found that for the same sampling period, November, the number of seeds and the number of taxa per sample was higher for El Rocío than for Hato Blanco rice fields. And for the same sampling site, El Rocío, we also found a higher number of seeds per sample and taxa per sample in November samples than in February samples. And here you can see the results from the brief movement analysis that we carried out, and we have the estimation of the places where seeds could come from and where they could be dispersed to from the sampling sites. And we can see that the seeds can fall in different areas, and in the last column of these tables we can see that this dispersal can achieve quite high maximum distances. So we could say that geese can be having an important role in the dispersal and connectivity of these plant communities, and that this dispersal can overcome the distances that these plants could achieve by other means of dispersal. In the second chapter, we want to assess the role of white storks in seed dispersal in Andalucía, identifying the plant species that they dispersed and quantifying alien and domestic plant species that we find, and assessing the effect of landfill use on the species composition found. So from previous works, from this previous work by López Calderón and colleagues, we know that white storks are using and connecting landfills with different croplands and also with natural wetlands like salines, marshes, and lakes. So we want to see how what effect this landfill use can be having in seed dispersal. For that, we collected 230 white stork pellets in 8 different sampling sites, 6 of them close to landfills. And the esa de Matagordas in the except of the esa de Matagordas and the esa de abajo. By now we have analyzed with the help of El Encañuelo 167 pellets, and we have found more than 1,400 seeds from more than 30 different plant taxa. So we want to see how landfill use can be affecting the differences in the plant communities that we found in these sampling sites. Then with the third chapter, we want to address which role can be having grey geese in the latitudinal long-distance dispersal of plants. This is a study that we will develop during the next year with Diego Pavón Jordan from the Norwegian Institute for Nature Research, and we haven't decided the final plan yet, but we would like to use geese tracking data and also plants occurrence data from citizen science platforms to develop some of the next ideas. We could do an estimation of the degree of co-occurrence between goose species and plant species recorded in their feces along the flyway, or an estimation of the degree of co-occurrence between recently arrived plant species at northern latitudes and geese flyways. Or also we could try to predict potential distribution of plants while accounting for changes in geese distribution and climate change. Well, and lastly, as I told you, the final step for a successful dispersal event is the establishment of the plants, and with this chapter we want to assess the effect that endozoochory by waterbirds have on plant establishment. For that, we tested the effect of gut passage and feces deposition on germination, plant growth, and seed production. This was an experiment that we carried out last spring in the Center for Ecological Research of Hungary, and we first went to the field and collected Juncus buffonius complex seeds in Doñana rice fields and Elocaris palustris seeds in EBD-CSIC protected area, which is a marshland in Hungary. And Raúl, in his talk, will explain why we chose these plant species. So with these seeds, we perform a gut passage treatment, feeding with 400 Juncus seeds and 100 Elocaris seeds, each one of the 8 mallards we counted with for the experiment. And then we collected the mallard feces in different time intervals for 24 hours, and we inspect the feces under the stereo microscope to extract the seeds. So for the sowing, we used these gut passage seeds and also control seeds, and to half of them before the sowing, we put a piece of mallard feces on top of the soil, and then we sow the seed on top of the feces. And here you can see the differences in the treatments, and here you have a summary of the number of seeds that we sowed per plant species and per treatment. Then we check the pots for new germinations daily during 1 month, and every 15 days we measured the plant height, and for Juncus we counted the number of flowers per plant. Then after 70 to 78 days after sowing, we collected the plants and we dried them to measure the above ground mass, and for Juncus we also counted the number of flowers per plant, the final number, and also now we are counting the number of seeds per fruit. So I can show you some preliminary plots because we haven't performed the statistical analysis yet, and here you can see the time to germination and the percentage of seeds germinated. And we can see that for Elocaris, there is a clear pattern where plant seeds sowed with feces in violet and green germinated less than seeds without feces. And in the case of Juncus, we cannot see such a strong pattern, but we can also see that seeds without feces in red and blue germinated more than seeds with feces. And in both cases, the seeds that germinated the most were the ones without feces, and with gut passage. In these plots, we wanted to see differences in the growth pattern between treatments, and graphically we cannot see a clear pattern, but it's true that for Juncus, feces could be hindering the growth, at least at the beginning, as we can see in these almost flat lines here in treatments with feces. And then regarding the above ground mass, in this case also, plants with feces had a lower mass than plants without feces. So all these variables seem to point out that feces could be inhibiting the plant establishment, and that gut passage can be having an effect, but that is not too strong to see it graphically. But we also looked at the investment in fruit production, and in this case we can see that treatments with feces, in this case in green and black, had a higher number of flowers at collection time in relation to the above ground mass of the plants. So we have to perform all the appropriate statistical analysis to see what is happening here with all these variables. So that's all. Thank you all for your attention, and thank you to my colleagues and people from the Aquatic Ecology Lab, and also from the last lab. That's all.
So suggestions, questions? I forgot to say that both the students are half.
Thank you, that was very interesting.
Thank you.
I'm curious that maybe there's some plants that are better dispersed than others, and I would also assume that there are other plants will change in how competitive they are, and there is a lot of at least hypothetical model saying that if you're very competitive, maybe you disperse less, and if you disperse a lot, you may be less competitive, so there is this competition dispersal trade-off. And I wonder if you know if for your system this is the case, or if you can look at it somehow.
Yes, thank you. And I don't really know. Like, we know that some seeds, for example, that small seeds can be dispersed better because they can survive the gut passage, maybe like escape the digestion, and they can survive better and be dispersed by the waterbirds. But the hypothesis that they could be escaping competition by dispersal, I don't really know. But thank you.
Thank you very much. It was a really interesting talk. I really enjoyed it. So, do I understand correctly that in the last part of your talk, that you followed the plants up until they flowered again, from seedling to, because you said you measured the number of flowers as a function of weight. So that was the plant you followed from seeds, or what?
Yes, they were this. Yes, this is the final number of flowers at the time we collected after all the experiment.
Okay.
Like, they were from this.
No, it's okay. Just to clarify that, that's correctly. Okay, now my question is, because when I see this, so there's the ones that were with the feces seem to be flowering quite a bit, so investing in reproduction versus and not so much into growth. So, is this suggestive of some kind of stress response, or because that is something that I would associate with, they don't grow so much, they flower as much as they can, because they're maybe stressed. That's my question.
Yes, that could be a reason. Like, we have thought about it, and this is the final number at collection time of flowers, and also the final mass of the plant. So this can be a stress, or it could be like we have thought in different things. For example, it can also be that the feces have some nutrients that boost the investment on flowers, and it can also be a stress because since they grow later than the plants without physis, maybe then they run to flower and flower more than the others. We don't know. We have to study it and read about it.
Are you following survival? Is it a long live? How long does the shrub live?
They were collected at between 70 to 78 days, but some of them were starting to die, especially the juncus, because they are annual species. But yes, and this is juncus, so that's the time.
We don't have microphones, so.
How you measure viability of the seeds? Because you say that you make germinability, but in which conditions you stratify it?
No,
or fold, or you use the tetrafolium, or?
Okay. We, like before extracting the seeds, the samples were in the fridge, so I don't know, they had some cold. And then we put them in filter paper, well, in plates with filter paper and in moist conditions, and that's how, and we wait them to germinate or not.
And you didn't do a second round of cold, and you didn't use tetrafolium?
No, that's not.
It's something, I know by experience that it's something reviewers used to request.
Okay.
That you really check that you are not dealing with seeds that are still dormant.
Yes, and maybe they could germinate if we perform that test. Yes.
Yeah. And the other question is, to what degree you consider that the treatment with phases is realistic? So
Yeah, I have thought about it because, like, for example, in the field, if a physis falls like in the ground, it will be more, I don't know, like erosionated because of the wind, of the rain, and here the physis persists like all the time in the around the seed. So yeah, maybe it can be like when it's no rain, no, but yes, it's different in the field.
because you are dealing with ducks that are living also in areas with lot of water and a lot of humidity. So in Beta La Palma, even without rain, the dropping from one day, the next day, it's in the way to disappear, and 1 week later has completely disappeared.
Yes, this is not like equivalent to field conditions. Thank you.
Is there any way to estimate the relative importance of waterbirds as seed dispersers compared to other dispersal processes?
I don't know. Really.
Because I guess these plant species can be dispersed by the wind, for example, right? For them to be dispersed.
Like, first, if we look at heavy seeds, that they are not supposed to be able to be dispersed by the wind, we could say that, like, if they are only dispersed by falling from the plant, maybe they cannot get to, and for example, these species that need moist, and they can grow on the sides of marshes or lakes. I mean, it's difficult that they can get another lake, maybe if they are not dispersed by an animal or maybe an extraordinary wind, but I mean, seeds adapted to the wind, to wind dispersal, maybe they can sometimes achieve great dispersal distances, but if they are adapted to just fall from the plant, I don't know. But I don't know how could we estimate the relative importance that waterbirds have.
I am asking this because perhaps you could estimate the genetic similarity between plant populations and if you also have data of the movements of the birds, you can perhaps compare the two networks and to see whether there is a match in the movements of the birds and the genetic similarity of the plants between these two sites.
Yes, Raúl, well, he's going to explain in his talk, but he's doing something like this with the population genetics. You will see.
Okay, so that was a very nice introduction for you, Raúl. So whenever you're ready.
Hi, and
Well, okay, so I'm gonna start. The second part of the seminar is titled The Role of Waterbirds and endozoochory in the establishment of plant populations and the connectivity between them. I am presenting here my thesis proposal that is going to be developed in the framework of the National Project Water Zoo, and my thesis directors are Andy Green and Maria Jesús Ortiz. I want to start the introduction with a definition of dispersal. That is that one. The voluntary or involuntary movement of an organism or propagule from its natural breeding site to a new one, where the establishment and the reproduction is attempted. With that definition of dispersal, we can classify two types of dispersers, the abiotic and the biotic. And if we think about the biotic ones, we would probably come to our mind the seed dispersal by any kind of bird. But these birds are now recognized as vital vectors of all these kind of organisms that are shown in the picture, and their dispersal capacity is the most ecosystem service that these birds can provide. In my case, for my thesis, I will focus on terrestrial plants. Okay, and on terrestrial plants, an important amount of research has been done on focus on the role of birds as dispersers of species. These angiosperms that have this fleshy fruit represents only the 8% of the total species in Europe. So what's happening with the rest of them? How are they being dispersed? If we search for that kind of information in flowering databases as baseflower, and we look for one of these species, Codula cornopifolia, we can see that the associated syndrome is anemochory, probably based on the wings that some of their seeds have. I mean, these syndromes are based on morphological traits only. But if we go to empirical data, as this picture from this paper that my colleague has explained before, we see that from the total amount of species found in feces from Dublin ducks, only the 8% of them had previously an endozoochory syndrome associated. So this kind of data is showing us that we need more empirical data to assess which is the truly dispersal syndrome of the different species. And with this empirical data, we can understand better the distribution of the different species, and that can be applied to conservation biology, restoration, or invasion biology. So with this background, the main question we want to answer in this project is if long-distance dispersal by a migratory waterbird is central to explain how plants move in response to climatic change, biological invasions, direct transformation of the habitats by human activity, or climatic change. And to answer that question, we will study the influence of the dispersal in the meta-community dynamics and in the meta-population dynamics. And for my thesis, I will focus on the second part, the meta-population dynamics. Now, I'm going to introduce you the 3 model species that we have selected for that purpose. The first one is Elocaris palustris, that is a Cyperaceae. The second one is Juncus euphonius complex, that is a Juncaceae. Both species are native to Europe and can be found in moist soil habitats, and they have been selected because they have high rates of waterbird endozoochory based on previous projects of the group. This is the distribution map of Elocaris palustris. We can see that it's widely distributed in Europe, North America, and dispersed in other parts. This is the distribution map of Juncus euphonius that has a cosmopolitan distribution. And here comes the third model species that is the buttonwood Cotula coronapifolia, that is native to South Africa and is being dispersed in an alien range in Europe, USA, and Australia. Here in the distribution map, if we enlarge to Europe, we can see that it's mainly distributed in coastal areas, but it's also spreading inland. Most authors assume that this is dispersed by hydrochory, but if we see the spatial pattern of spread of this species inland, and we compare them to the movement of waterbirds, of one waterbirds as godwit, we can see that this more consistent with this with a dispersal by endozoochory. So here's another example where we can see one of those inland wetlands in UK, Wirral, that is crowded of godwits, and also it's full of Cotula. And in this paper, we can see that they extracted seeds from this species, from Cotula, or from faces of those godwits, and from all the seeds that they found in the faces, more than the half of them finally germinated. So it demonstrates that this species is being dispersed by endozoochory, at least in Europe. Coming back to Spain, I have to say that this species has also registered a certain salt tolerance, and this could explain the colonization of the Doñanas marshlands during the last drought periods. So now, once that I have presented the topic of my thesis, I'm going to start with the plan of the different chapters. The first chapter is titled Germination Pattern and Establishment of Cotula coronapifolia after the good passage. And here we have the objective to investigate the establishment success of Cotula coronapifolia seeds dispersed by waterbird endozoochory. And we have divided it into experiments, a germination and an establishment experiment. For the germination experiment, we ask mainly 2 questions. How does the good passage simulation affect to the germination of the seeds, if positively or negatively? And if the salt tolerance of the species allows to colonize more saline habitats. For that, we simulated the good passage to the seeds of Cotula, and then we placed them in Petri dishes with agar and 4 different salt concentrations that were 4, 0, 5, 10, and 15 grams per liter. Then we put the plates in a germination chamber during 20 days, and we recorded the germination of the seeds daily. I'm still analyzing the data, but here we can see some preliminary results in these 2 plots that shows the cumulative germination of the seeds in 2 of the populations that we analyzed. the left one coming from Cádiz and the right one coming from Doñana. And I just want to say that in both graphics we see in red the cumulative germination of the seed of the control seeds in 10 grams per liter of salt, and the blue line indicates the cumulative germination at the same salinity of those seeds that had the good PAISAJE simulation applied. And as we can see in both cases, there are significant differences between the treated and the not treated, and this is indicating that this simulation affects positively to the germination pattern, at least with these species. Now for the second experiment, the establishment one, we ask this question: how does the good PAISAJE simulation affect to establishment of the adult plant? And for that, we will plant control seeds and seeds with this good PAISAJE simulation, and we will subdivide them in different groups that will be water with normal water and different salt solutions, so that when the plants are grown and in flower, we will measure some physiological parameters as the plant height, dry weight, total chlorophyll, or anthocyanins. So from this chapter, the first part is done and the second part is in progress. Here comes the titles of the second and third chapter that are titled Population Genetics of Cotula cornopifolia and Elycocaris palustris. And both chapters has the same aim, that is to determine and explain the population structure of alien and native plants with high rates of endozoochory by waterbirds. The workflow for both species was first the population sampling, and for that task we have a group of international collaborators that have helped us to obtain samples from different populations along Europe and South Africa. This map, in this map I show the population sample in Europe for the first species, Cotula cornopifolia. In red there are the populations that we selected, and in green the discarded. We had to choose the populations we wanted to analyze, so the selection criteria was having enough material for the analysis, having a representation on each country of coastal and inland populations, and having enough distance between the populations to ensure enough variability between them. Here it's shown the selected populations in Portugal and Spain. We only had to discard 2. In UK the same, we only had to discard 2 populations. And in the Netherlands, Germany and Sweden, we decided to discard the Germany population because we only had 1. In South Africa we have 5, and we finally selected 3 of them. This table sums up the final number of populations that we are going to use for the analysis, the number of individuals per population, and we try to maintain a constant distance between the individuals on each population. This map represents the sample size for the other species, that is Elycocaris palustris, and for this species we didn't have a selection problem because we could include nearly all the populations, but we need again enough material, and for this we selected natural and artificial habitats on each country. This table sums up the total amount of populations, the number of individuals, and compared to Cotula, for this species we have an extra population of seeds grown in a greenhouse coming from feces of waterbirds. The second step was the DNA extraction that was done with a commercial kit with some modifications, and then we have to fill the plates and send them to a company to do the library of the GBS and the GBS, that is genotyping by sequencing. For those who are not familiar with this technique, it's a technique that digests the genome DNA in our case with 2 enzymes, then these fragments are ligated to some adaptators. Then the fragments with adapters are amplified by PCR, and then the products of the PCR are sequenced. But why did we choose that technique? We chose the technique because we wanted to use SNPs as a source of information of variability between the populations. The SNPs are single nucleotide polymorphisms, and this variation, single nucleotide polymorphism, that varies in one nucleotide in the same sequence within the individuals of a population. And this variation has to be present in more than 1% of the population. If not, is considered just a punctual mutation. Secondly, because it's cost effective due to our objectives compared to other techniques that allows us to obtain the same SNPs. And thirdly, because we don't need a reference genome to obtain them. In this table, we see that using that, we will be able, without a reference genome, to obtain tens of thousands of SNPs, depending on the pipeline we use and the bioinformatics. And with this information, we want to do something similar to this. That is the population structure and a neighborhood joining tree, for example, to therefore be able to assess if the population structure and gene flow of the different populations is consistent or not with a key role of Waterbird endozoochory. So the first step is done, the DNA extraction is done, and now we are preparing the plates to send them to the company. And finally, for the fourth chapter, is titled Disentangling the Juncus-Euphonius complex. In this chapter, we have the aim to determine which taxons from the Juncus-Euphonius complex are being dispersed by waterbirds and which is the connection between them. This complex is formed by moreover 6 cryptic species that are divided in 3 ploidy levels, the diploids, the tetraploid and hexaploid. And the problem is that they are difficult to determine. When we find a seed in a thesis or we grow the adult plants, it's really difficult to get the species just with the morphometrical data. So well, we collected samples along Europe of this complex of species, and the first thing we did is to measure the ploidy level of all the samples. And as preliminary results, what we can see in this table, and what caught our attention is that from the natural habitats in Spain that comes from Doñana, Laguna Dulce and El Martinazo, we can see that the 3 ploidy levels are mixed in the same populations, but in the same area, in artificial habitats like the rice fields in Puebla del Río or in Puebla del Río, Isla Mayor, we found only 1 ploidy level that was the diploid one. These data were taken in a short stay in Vienna with Karin, and apart from that, we also tested some molecular markers with very few individuals to see which was the connection between them. And we found that the rice field population, the ones that were homogeneous and diploids, was the most distant ones compared to the other populations of Netherlands, UK, and Doñana. So to assess this objective, we are going to use molecular markers, ploidy level measurement, and probably we will use a phylogenomic technique that is called HAE-Intro. And that was all.
Suggestions, questions? Raúl.
Festin talk. How do you do the gut passage simulation?
I did the gut passage with 2 steps. One step that was scarification with some grit, with some grits and water during 2 hours, shaking in a machine at 37 degrees. And a second step that was adding chlorhydric acid with pH 2.5, another 2 hours at 37 degrees.
Interesting, thank you.
Well, first of all, thank you for the talk. It was really nice. And I just wanted to ask what is haploidy level, because, for example, I do not work with genetics, so I got kind of lost in that part. If you could clarify it.
Is the number of copies of the genome of an individual. In this case, we have the exaploid individuals have 6 copies of their entire genome and so on.
The seeds of Juncus are very small. And do you consider the possibility that the seeds in the rice fields are pollution in the seeds that the farmers are using when they put the rice?
Well,
you understand what I mean?
That
you go, the farmers go to the provider and they got a big bucket of rice seeds, but in these seeds you don't only have rice, you may have
also Juncus,
other plants. And one possibility is that these seeds that they buy are polluted with seeds of Juncus. So maybe it's a good idea that you check in some seeds provider if you can find seeds of Juncus in the mixtures they are using for seeding.
Okay. I mean, yes, it should be interesting to check that. Because maybe it's not because of Waterbirds, because of human activity.
Yeah, because they are taking the seeds from Valencia or from Asia. I don't know where they are producing the seeds, but it can explain why they are so different.
Okay, thank you.
What is the genetic structure that you expect from endozoochory plant species? Because you say that studying the genetic structure of the population might indicate whether they are endozoochorians or not, like for that plant species.
And the genetic structure that I
you expect that you might find
well,
in the population.
I expect that the population structure is more similar between those populations that are matched by a migratory route than those populations that are not in this line.
And are you gonna also check the genetic structure within the population? So
yes,
how?
That's what we plan, but you know, I don't have the data yet.
Okay. Any more? Okay. Anyway, they are PhD students. They are happy to get more feedback. So if you have any more suggestions or questions, let them know afterwards, after the seminar. Okay? So thanks a lot both, and thanks all of you for coming. Thanks.